Welcome everyone to the webinar on anaerobic soil disinfestation as an ecological tool to regenerate soil health in specialty crop production systems by Dr. Jaya Nepal of Penn State University. I'm your host, Alice Formiga of eOrganic. This presentation will last approximately 50 minutes, and then we'll have another 30 minutes for audience questions. This webinar is being recorded, and you'll be able to find the recording within the coming week on the eOrganic YouTube channel and the eOrganic website, along with many other webinars about organic farming research. You can also use the chat box if there are any resources that you'd like to share with the other attendees. We have conducted several webinars on anaerobic soil disinfestation over the last few years as part of the NIFA OREI-funded project ASD Easy, led by Dr. Francesco De Gioia of Penn State University, who's going to be online with us for the Q&A today. So today, I'd like to introduce Dr. Jaya Nepal, who's a postdoctoral scholar at the Vegetable Crop Sciences Laboratory at Penn State. Jaya has a PhD in soil and water sciences from the University of Florida, where his dissertation focused on evaluating the effects of amendments like biochar on soil. In his current role, he's studying the effects of ASD on soil health. Welcome, Jaya. I'm going to turn off my video here, and you can go ahead.
Yeah, thanks, Alice. Thank you, everyone, for tuning in. It is my pleasure to present today on ASD. Particularly, we've been working in this project for almost 5 years now, and with ASD for almost a decade. Led by Dr. Francesco Di Gioia. So, especially in the beginning, we worked a lot with ASD as a disease management tool, but over the years we've refined our protocols, systems, and really been more focusing on overall system and soil health rather than just as a biological tool for disease management. So we have about four years of results on soil health, which I'm going to present. Like Alice mentioned, this is a part of a webinar series coming from our ASD Easy project, and we've covered a lot of ground, particularly in discussing ASD, how does it work, different mechanisms, what are some of the amendments or carbon sources you can use, and what are the different rates you should be using depending on system. We also discussed about how we can sort of incorporate cover crops. And last year we also talked focusing on how can ASD be applied in high tunnel systems. And this part really is going to be focused on using cover crops as well as organic amendments for ASD and how it affects soil health. Like Alice mentioned, this is a part of a multidisciplinary, multi-regional project, and we've been doing work in uh, Pennsylvania, but also in Florida, at University of Florida, and also USDA areas. And we've been working with vegetable crops as well as fruits, especially strawberry in Florida, tomatoes, and other vegetables in Pennsylvania and also in Florida. And we have a really interdisciplinary team looking at the ASD optimization, soil microbiome, soil health, pest management, and also have extension component where we've done on-farm work, and we aim to do a final technical economic analysis, which could be an upcoming webinar, and we'll keep you posted on that. So coming back to today's content, I will start with what are some of the challenges, issues with specialty crop production, and how we can apply ASD as a strategy for building soil health in these systems. How can we take advantage of incorporating cover crops for building soil health, and also how can we complement cover crops with organic amendments such as wheat middlings? And I will really share our results from soil health that we've done in the past 4 years as a part of ASD EG project, and this is coming from a 4-year field work in Pennsylvania, working with ASD and tomato in an open field. Vegetable production systems are inherently intensive systems, particularly if you're growing multiple crops per year or you're growing same crop year after year. Soon enough, you'll start to notice that you're pushing your soil to a biological limit, and then different problems start to prop up. Specifically, a lot of growers have issues from soil-borne pests and pathogens. Uh, once you start to grow same crop in a soil for year after year, the pathogens sort of develop and thrive over time using the same host. And as soon as they get a favorable environment, different fungal, bacterial, and nematode start to affect the crops. And some of the examples that we see typically are verticillium, fusarium wills, coleotrichum, different rods, black rot, and also nematode issues such as root rot nematode. We also have dagger nematode and different nematode issues. To make it more complex, there are also issues with the weeds, and if you don't manage weeds, that sort of feeds into the pest and pathogen, and it forms sort of like a even more complex problem where you not just have one particular issue, but a lot of these issues are combined, and we typically see, let's say, a fungal and a nematode problem sort of intertwined and connected together, which makes it really challenging to control or manage these situations. In addition, there are issues with nutrient, soil fertility, issues such as salinity, and if irrigation is an issue, issues such as blossom inrot, different kinds of nutrient imbalances, pH issues, and really a systemic soil health degradation starts to happen over time in these systems. And this is true for both open field as well as high tunnel productions, and we've worked in both of these systems here in Pennsylvania, with both with growers and also in our research fields. Looking at a bigger picture globally, these plant pathogens, nematodes and weeds, they cause about 20 to 40% global crop loss annually. And this is really a big bottleneck for specialty crop production here in Pennsylvania, but also throughout the world. And if we don't manage these early enough, and if we don't build resilient systems, pretty soon we start to see complete crop failure where a farmer or a grower cannot even grow a crop uh, in the following season. So this is pretty damaging issue throughout the globe. And I know that a lot of people today here are probably coming from an organic management system. So this methyl bromide was a one-stop fumigant that used to be applied across different systems, especially in commercial conventional systems. Now, this was never an option for organic systems, but what it did is once people started to realize that this was a fumigant that was affecting the ozone layer and it was slowly phased out, a lot of interest started to work towards finding a solution that is sustainable, that is biological, and that can be applied across conventional as well as organic systems. So the method that we're talking to today, anaerobic soil disinfestation, that came about in early 1998 or early 2000s. So a lot of drive that came from was really from the banning of this chemical or this one-stop fumigant, which really pushed scientists and researchers to find a solution that can be applied that is ecologically sustainable. So Since the phasing out of this fumigant, there've been research in other chemical fumigants, and some of them are currently available in conventional systems. Some of them are being tested, but really replacing one chemical from to another, we believe is not really a viable alternative. And with increasing pressures on the environment, regulations, sooner or later we'll have to find sustainable, systemic, and biological solutions. Uh, and anaerobic soil disinfestation is one of many solutions that can be applied biologically. Some of them are steaming or steam treatment using biofumigation, biocontrols, solarization, biosolarization, and so on. And often you don't even rely on one method. You can even complement different methods and also use cultural methods such as crop rotation, cover crops, biochar, and different other things to sort of combat and make the system more resilient to disease and soil-borne pests. So anaerobic soil disinfestation, if you don't know about this, it is also sometimes called as biological soil disinfestation or reductive soil disinfestation. So it is really a biological strategy where we create an anaerobic condition in the soil for about 3 to 4 weeks. And what that does is it promotes the anaerobic microbial community in the soil, which produce different organic acids. It produces volatile compounds and really improves the suppressive ability of the soil. So it improves disease suppression significantly, but it also prevents weed, and also it contributes to building soil health over time. It is essentially a three-step process where we first amend the soil with an organic amendment. So here you can use different amendments. The best is if you can find an amendment that is relatively labile and that can be acted upon by microbes immediately after applying. So even if you're applying something, trying to grind it or putting it in a smaller surface area is going to have a better result. And also another thing to note is the CN ratio, because it's a microbially driven process. We need to maintain a certain CN ratio that I will talk later in the show, but around 20 to 1 carbon to nitrogen is where we find the best results with AST. The second step is to mulch. So essentially, once you applied organic amendments, you cover the soil surface with an impermeable film. So the idea is to prevent the exchange from the environment, and you create a completely anaerobic conditions. Then you apply irrigation. Typically, we say apply irrigation to field capacity, so around 2 to 3 inches, and you only need to do it once. So once you create an anaerobic conditions and you have this mulch to prevent external oxygen, the anaerobic microbes really start to feed on these amendments and produce those organic acids, volatile compounds, and really create an environment that can suppress disease, pests, and weeds. Uh, So, before going into the field study that I'm going to talk a lot about today, I wanted to briefly share the work we've been doing on farm. So this was a organic farmer who had issue with a dagger nematode. He used to grow raspberry, and essentially he could not have any crop. So he reached out to us through an extension agent, and we found that he had I believe, like north of 10 times the nematodes in that soil. So we started working with the grower. We applied organic amendments, and as you can see, he was an Amish farmer. So it also shows that we can really apply this technology at a small scale, even using the local resources. As long as you have an organic amendment, you can irrigate the water and make it saturated. Here we were testing two different types of impermeable film, clear plastic, and also black one. Uh, we found that both can be used to create that impermeable layer. And after we re-aerate the soil, we also we did some tests, and we found that AST was able to kill most of these dagger nematodes. And I think the farmers, we recommended the grower to grow a short season crop before going into a full raspberry because it's a perennial crop. So we wanted to see sort of a longer term effect. And once I think he grew cucumber next, and then once we did, we evaluated the soil again and found again that the dagger nematode was in control. He went with the raspberry, and we could see that he really had good raspberry production this year. So this is an example. I wanted to share this not only to show that this was kind of a success story in the farm, but also sort of to point that this was a case where there was an issue and we acted upon an issue, and we could, even though we saw that ASD did work, it is kind of like a reactive management. So, and then we started to think about why don't we manage the soil proactively? So, not just like we go with ASD when we have a particular issue, and typically when the issue is really complex, but rather than treat it as a systemic tool to improve soil health that can generate or that can produce suppressive soil that can enrich the soil with fertility and also really improve the soil physical, chemical, and biological properties. So we started to think along the lines of how can we combine ASD with soil health principles. As many of you might be aware, soil health is really rooted in four ground principles to maximize soil cover. So growing cover crops is one of the options. Keeping the soil covered throughout, even you are not growing the main crop, is a key principle. Maximizing living roots, really rotating your crop, adding diversity to maximize the roots, because roots play an important role in the soil to create the soil microbially active and also biologically suppressive. Another principle is to maximize biodiversity. So, by adding different crops, by rotating crops or adding cover crops, we can really maximize biodiversity in the soil. And another principle is to minimize disturbance. So, don't till as much as you can. We know that in vegetable production systems, tillage is sometimes a necessary evil. We have to till sometimes to create bed and all those things, but try to minimize it as much as possible to build soil health. And you can also integrate livestock, which can be done in this system by adding manure, chicken manure, for example. So really, and we started to look into different cover crops and why we should use cover crops. And yes, cover crops add a lot of above ground biomass, which can be a source of carbon in the soil. But really, the roots of the cover crops are really creating an environment in the soil that is releasing a lot of root exudates, a labile carbon source that microbes can feed upon and become really active. So we started to investigate different cover crops, and we also investigated how we can integrate cover crops as well as different amendments. So here in this experiment, we were looking at wheat middlings, cover crops, chicken manure, molasses, so a bunch of different organic amendments, and really trying to explore how we can capture the synergy of cover crops and organic amendments to build a resilient system that has a better soil health. So, like I mentioned already, ASD requires a labile carbon inputs. This is because if we don't have labile carbon inputs, it will take time for the microbes to act upon, which will mean that we'll have to sort of extend the ASD periods, which we don't want. We want to typically do it in 3 to 4 weeks. So we've done work with different kinds of substrates, and uh, like all of them have different pros and cons. For example, if you use an apple pomace or spent mushroom substrate that have higher moisture, even though it is, you cannot still apply them, but you have to sort of immediately apply and you cannot store. Wheat middlings, for example, on the other hand, you could uh, get get whenever you want, and you can store them for a longer time. And we also found that this has a CN ratio of around 18:1, which was very close to the optimum ratio that we need for ASD. So we have explored different organic amendments, and really what amendment you can use depend on a bunch of things. So like I mentioned, composition, CN ratio, but also availability, cost, and other things. So some of them can be costly, especially if you have to import from a distance. So the delivery can be challenging. So all of these factors need to be considered in deciding which organic amendments you can use. So the way we can sort of reduce that factor is by producing something in your field so that you don't have to rely as much as on external inputs such as organic amendments, and you can grow cover crops, which, like I mentioned, maximizes soil cover, maximizes living roots, and also helps building that diversity. But it also provides an opportunity to produce a low cost on-site carbon, and if you mix a legume, also a nitrogen source. Uh, and some of the agencies, such as NRCS, offers a cost share program to grow cover crops, and it can be really integrated well in the organic systems or soil health systems. So that was another motivation in using cover crops or testing cover crops' effect on soil health. Like I mentioned, it reduces the need for organic amendments. And really, we can tap into the synergistic advantages of both cover crops as well as complement that with potentially different organic amendments. So we started looking into different cover crops. This is a study from different research stations as well as commercial farms across Pennsylvania, conducted by Kathleen and Jason Kaya lab here at Penn State. So we looked at different leguminous cover crops. Typically, these provide nitrogen source, so it adds to fertility and it supplies nitrogen, but it doesn't supply enough structural carbon, which is sometimes needed for ASD, and also it is not as efficient in suppressing the weeds compared to non-legume cover crops such as triticale, for example, uh, which has a higher CN ratio, which means that there could be a potential nitrogen tying, especially early in the crop stage. But really, if we can, what we found is if we can mix them up, especially legume and non-legume, you can really fine-tune the cover crop carbon to nitrogen ratio and really have a good control of weeds, have a good structural as well as labile carbon source, but also add some of the nitrogen, so that will reduce the need for fertilizer later down the line. How can you incorporate cover crops? Really depending on where you are and what is the crop rotation you have. We are here in Pennsylvania uh, where we have to grow overwintering cover crops because the winter you really don't grow a lot of cash crop. You can extend it by using hide tunnels, but typically what we do is grow overwintering cover crops, and then in the early spring or summer, we go with ASD for about 3 to 4 weeks, and then we grow summer grass crops, which could be vegetables or any specialty crops. This is the second one here is the case of Florida, where the strawberry is grown in from August to all the way to April, and then you can, after harvesting all the strawberry, you can go with a summer cover crop. This could be a sun hemp or a Sudan grass cover crop or a mix, and then you can apply ASTV for another cover crop. So depending on where you are, what kind of system you have, you can grow different kinds of cover crops. as a source for carbon in AST. And like I mentioned, cover crops also provide other advantages. Uh, I will focus to our system. So here we seed the cover crops early in the fall and we let it overwinter through the winter. And typically, when it's around 50% flowering, then it is the good time to sort of go in with flail mower. So we typically do 2 to 3 rounds of flail mowing to chop the cover crops, and then we plow and disc the cover crop so that it is incorporated well, and then we create a soil for the next crop. And typically at this stage, you can apply other organic amendments such as wheat middlings or any other amendments that we have. So with that, I'm going to sort of talk about the study that I will be showing results. So this is an certified organic tomato production systems at Penn State, and we conducted a field study with a split plot design where cover crop was our main factor. So we had four different, including a fallow control. We had a triticale, which is a grass cover crop, crimson clover, which is a legume cover crop, and a mixture. We also had an untreated control, which I will not go into a lot about in this talk, but we will treat this fallow as sort of like a control without cover crop. And what we did is with the split plot design, we terminated the cover crop, and half of that we added wheat middlings, a complementary organic amendment, and half of that we only had cover crop. So we really wanted to see if cover crop alone was sufficient as a carbon source, or it is probably better to complement still with a more labile source of organic amendment. So the AST and the tomato cycle was repeated for 2 years. 21 was when we started, and 2024 is when we did the final sampling. And we did ASD for 4 weeks, and this was repeated 2 times in 2 years. And each time after ASD, we grew tomato crop and then went into a overwintering cover crop. So we grew Red Deuce F1 as a tomato variety. And for the in-season management, we didn't apply any additional fertilizers. So after cover crops and organic amendment, where we didn't apply any other fertilizer. We used soil moisture sensors for irrigation, and we didn't do any weed control. Uh, Again, like I mentioned, we had crimson clover, triticale, and mix, and we had a fallow plot, and we splitted these into plots having wheat middlings and plots without wheat middlings, and we added water to create an anaerobic condition. And then in half of them, we added wheat middlings, which is another organic amendment. And in the untreated control, we had a fallow and we didn't add any water to that. So the timeline of the experiment, like I mentioned, we grew cover crop through the winter. We went with ASD for 4 weeks, and then we had tomato for 2 years. And then this was repeated in 2021, 22, and 2023, 2024. So moving on to the results, I will present cover crop biomass, and then I'll pause to explain a little bit about soil health and soil health indicators, and then I'll continue to nutrient dynamics, and I'll also present some of the results from tomato marketable yields. So like I mentioned, we had triticale, which is a grass cover crop here. We had clover, which is a legume cover crop, and we had a mix. And what we found was that when we had a grass cover crop, it was typically higher biomass compared to a legume only cover crop. So even when we had triticale only or triticale with a crimson clover, we produced a similar kind of biomass across years compared to crimson clover, where there was a lower biomass. And what is also interesting here is that whenever we had grasses, there was a much better weed control. If you look at the triticale cover crop or even the mixer, we could see that there was almost no weeds. other than the cover crops. Whereas when we had a crimson clover monoculture, we had weed issues, especially in the first year. So the selection of cover crops really not only affects the soil health that we'll discuss later, but weeds is another important factor to consider when deciding which cover crop you want to grow. We also found that other than the biomass production, what time we terminate the cover crop is also important because as we delay the terminations, which happened to us in the second season because of different issues, we had a really wet winter, so we couldn't terminate the cover crop with, sorry, spring. So we couldn't terminate the cover crop. As a result, the nitrogen concentration was lower. So, like I mentioned before, Triticale had around 25 to 28 carbon to nitrogen ratio, whereas Crimson Clover had 12 to 15. And we saw that a mixture was around 18 to 20, sort of intermediate. And this is where we sort of think that a good combination of labile and less labile carbon source that can provide structure, but also can feed the microbes so they can produce those organic acids, volatile compounds, and really uh, make the soil anaerobic in a short amount of time. So for the soil health, I should mention that we took a sample before starting our experiment, and before actually we went uh, with our crop. The field was a complete grassland almost, so that was sort of the field converted from grassland, and then we took a sample there, and then we took two samples in 2024, and we took three samples in 2024. So for the ease of today's talk, I will mainly focus on a post-harvest sample from 2022, so after the first season and after the second season. And in some of them, I will summarize these two and these three to see really the overall impact of cover crop and organic amendments. We measured a bunch of soil health indicators. We sent our samples to independent laboratory, which did the analysis for us. We also did some of them sort of to verify at Penn State, uh, but yeah, we measured different soil health indicators, and I'm going to present some of them today, and hopefully try to explain some of them to you. And we also had overall soil health and soil fertility scores across the treatment. So before going into the results, I wanted to point out that, like I mentioned, like I showed here, there are like different kinds of indicator you can measure, and there's always sort of the balance between how much you can measure. And Soil Health Institute as well as USDA really recommends uh, to to measure at least three important parameters. So soil organic carbon is a really important parameter. Soil aggregate stability is a physical parameter that is very important, and 24-hour carbon mineralization potential or soil respiration. So if you are measuring at least these three set of indicators, this you're doing sort of the basic, or you're capturing a lot of the changes in soil health with your management systems. Why these parameters? So these were selected because these are the most responsive to management. This can be also done at a larger scale, more economically, and it links really well with soil function and ecosystem services. So soil organic matter influences, it is really central to the soil health concept. So it affects soil biological, chemical, as well as physical properties. And it also affects soil fertility, nutrient supply, crop yield. So even though it varies in the kind of soil you have, typically we say that as you increase soil organic matter by 1%, you increase the yield by about 10%, and you also increase the water holding capacity of the soil. So you can measure overall soil organic matter. You can also measure an active fraction, which is also known as a labile organic matter or active carbon. So this is sort of an important soil health parameter that we're looking at. Another one is soil aggregate stability. So this is really telling you uh, the ability of your soil to form stable aggregates. So when you have more stable aggregates, then it is more resistant to breakdown, especially from wind erosion, water erosion, and also it is related to infiltration, water holding capacity, and also it affects the soil microbiome. So it is good to have a higher aggregate stability. And 24-hour carbon mineralization potential or soil respiration really measures uh, how microbially active the soil is at a given point of time. It is also a key indicator, and higher values typically indicate a larger, more active microbial community. And even though we'll not go into really detail on which microbial community and structure they were, we could see a difference in soil respiration at different sampling we've measured. So with that, a little bit of information to put things into context, I would like to go into some of our results. So soil organic matter. So I'll present most of the data in two different kind of structures. So this is one of the structure where I have results from 2022 and 2024. And on the left here, we have effect of amendments. So the red one here is on a no wheat middling amendment. The green one is wheat middling with wheat middlings. And on the right here, the gray one represents no cover crop, and the orange one represents a cover crop treatment. And the sort of dashed dotted line is the baseline sampling that we did before starting our experiment. So this is sort of the baseline that we're comparing our results against. So for the soil organic matter, we didn't see a significant effect of organic amendment that we had, even though we could see slightly increasing trend compared to no amendment that we expect. But really, we started to see more significant effect with all the cover crops. And we could see that throughout 2022, as well as at the end of the 2024, we could see that a cover crop treated plots has significantly higher soil organic matter percent compared to the plots without cover crops. And we could see that over time. So compared to 2022, if you look at here, we could see that even compared to our baseline sample, the organic matter seems to be steadily increasing, especially with the cover crop treatment. So that was really encouraging. And here, this is the kind of second type of graph where we have a cover crop in the x-axis. So we have fallow, triticale, crimson clover, and mixture here. And we have wheat middlings here and without wheat middlings. So this is cover crop without wheat middlings, and this is cover crop and wheat middlings. So this is really showing a relative, the color is showing the relative differences between these treatments. And we could clearly see that compared to a fallow plot that didn't have any cover crop or wheat middling amendment, we could see that when we had cover crop in the system, it resulted in higher soil organic matter. So the roots from the cover crop that we talked about, the biomass, is really helping the soil to build organic matter over time. And we could see that with, especially if we complement with middlings, we could see that it also helped to build soil organic matter in time. But if we only had with middlings, there was no significant improvement in organic matter over time. This is the active fraction of carbon. So this is something that we expect to change relatively quickly compared to the organic soil organic matter. And this is done through a permanganate method. So epoxy. So really it tells about how much available carbon there is in a given time that can feed the soil microbes. So it is really a good indicator of how your management is affecting carbon in a short time. And here also we didn't see effect of amendment, but we sort of mirroring the soil organic matter. We saw that in our cover crop treatment, we saw an increased active carbon whenever we measured active carbon throughout the cycle. And when we look at different cover crops, we could see that really the leguminous cover crop, the crimson clover, was performing the best at any given time, and it had the highest amount of active carbon whenever we sampled. And compared to, let's say, a fallow plot or the plot only treated with organic amendment, all the cover crop treatment had a higher active carbon at any given point of time. So we could really see an effect in organic matter as well as active carbon. Next, we're looking at an aggregate stability. So here you can see that because we are sort of tilling the system to incorporate the cover crops compared to the baseline, we are uh, sort of over time reducing the aggregate stability of the soil compared to the baseline. But if you look at uh, the organic amendment as well as cover crop, we see a trend that when we have those amendments and cover crops, it is not sort of declining the aggregate stability as much. And especially by the second year, by the end of the second cycle, we could see that when we had cover crop, it had a significant increase in aggregate stability compared to when we didn't have any cover crop in the system. So cover crop, as we expected, was improving the soil aggregation, and it it also correlates quite well with organic matter and also the microbial activities that we're seeing. And when we look at different cover crops, we could see that triticale cover crop, which is a grass cover crop, which produces, I would say, the most dense rooting system, was promoting the aggregate stability the highest compared to a fallow plot, followed by a crimson clover plot and a mixture, so which was sort of intermediate. So this was sort of what we expected. And soil respiration, here also you could see that compared to when there was only a grass-based sod in the system, the respiration was higher because of the mineralization of organic matter through tillage and through cropping system. But we could see that compared to both no wheat middling as well as no cover crop, both of our amendments, cover crop as well as wheat middlings, improved uh, or showed a significant higher microbial activity at different sampling time and overall in post-harvest samples of 2022 as well as 2024. So we could see that when we incorporating organic amendments and cover crop, we were improving the microbial activity of the field. Again, looking at different cover crops here and amendments, we could see that sort of mirroring when we had wheat middlings in the system and also cover crop in the system. It was much higher compared to when we didn't have any cover crop or any organic amendment in the system. So typically, I think we are still in the, let's say, lower range of microbial activity. So around 200 to 600 is typically considered a good microbial activity. So that could be something we could be looking at potentially adding a biostimulant or bioinoculant or any other amendment that can further improve the microbial activity in this system. So that is something that we haven't looked at, but we plan to look in the future if we can. So this is the relative soil health score. And here I'm showing only the cover crop and wheat middling. So we could see quite clearly. So this is sort of capturing all the different indicators that feeds into this 0 to 50 scale. And we could see that compared to no cover crop and no wheat middling treatment, which was kind of like a standard or business as usual, we could see that especially when we had cover crop in the system, our soil health was building over time, and this was reflected in our overall soil health score. I do need to mention that this is still not, let's say, an optimum soil health range that we want to achieve. So we are still looking at different ways we can sort of accelerate this building. But as you might know, the building soil health overall in the system does take some time. So we will continue to monitor this over time, and we do expect that over time our soil health will improve, will continue to improve. And if we look at different cover crops here, we could see that when we had a triticale and also crimson clover cover crop with middling. So typically, whenever we could see that when we complement cover crop with a labile source, in this case with middling, this is when we are seeing a more sort of accelerated increase in soil health compared to when we had only cover crop, where we do see an increase, but it's not as as rapid as when we have both a cover crop and organic amendment in the system. Uh, so with that, I will sort of pivot more into soil nutrient dynamics. Uh, Again, this is also part of a broader soil health, which accounts for physical, chemical, and biological properties. But I will, for the ease of this talk, I am putting them into sort of more into the chemical and nutrient part. So, in terms of pH, we didn't see any real impact of different cover crops and organic amendments. So all of these pH were sort of in an optimum range, which is like typically 6.5 to 7 is considered optimum. So there was no significant effect on pH, even though we did see some changes during the AST phase, but overall the pH didn't change that much. Soluble salt or electrical conductivity. So this reflects the total amount of soluble salts. And if we look at this, even though we do see sort of increase in soluble salts, uh, we are still in the range. So this is, let's say, zero point 0.1 to 0.15. So below zero point 0.2, it's still kind of like low salt level. So we don't need to sort of worry about any salt buildup or anything. It really indicates that there are more sort of ions in the soil, and that can provide nutrients to the crop. So it's only if we start to go, let's say, in more than zero point 0.8, then we start to maybe worry about salt buildup. So in both of the cases, we saw that with amendments uh, and cover crops, so we were having a greater increase in soil electrical conductivity. And in terms of like different cover crops, we didn't see a clear difference here. So it was sort of in the similar range here. Cation exchange capacity, which is a measure of the amount of positively charged cations that the soil can hold. This really depends on the kind of the type of soil you have. Sandy soil, for example, typically have this in the range of 0 to 5, certainly below 10, as opposed to a more fine texture soil where I would say around 10 to 20 is considered sort of a good level. So we had a good level of cations throughout the experiment, and we saw with crimson clover, which is a leguminous cover crop. So this was supplying a lot more cations in the system compared to other systems, but they all had a sufficient level or an expected optimum level for the loamy or fine texture soil that we have here. In terms of soil fertility, here we are looking at plant available nitrogen. And we could see quite clearly that in when we add organic amendment in the systems and also cover crop, typically, especially with the crimson clover or mixed cover crop, we could see an increase in soil nitrogen, which is sort of the plant available nitrogen. And this was also reflected when we did a plant tissue analysis and also tomato nutrient analysis. We could see that this nitrate level was actually higher in different parts of the tomato. So this was reflected throughout our AST study. We didn't see a difference in terms of soil ammonium, even though we saw that, especially during the AST phase, so during the first 4 weeks where we actually do have the ASD, there was a slight spike in ammonium, but over time we didn't see a significant increase in soil ammonium levels across our study. When we look at different cover crops here, so as expected, we saw a higher level of soil nitrate with crimson clover. So as you expect, the crimson clover was adding nitrogen in the system, and the mixture provided some of that also in the system. In terms of soil ammonium, we didn't see a significant change. These values were quite low. So even though there seems to be some fluctuations, I think some of that is related to some of the ammonium being tied up early in the with the triticale, and later we didn't see a significant difference in ammonium levels. Nitrogen mineralization, which is an indicator of how much nitrogen can be released from organic matter during the season. So this is where we saw a clear advantage of a crimson clover cover crop, especially when complemented with wheat middling. So this was showing that whenever we had crimson clover, there was more. The soil was able to supply nitrogen not only for that season, but also there was some residual nitrogen for the coming seasons. And we could see that in different studies. So not only this study, but some of the studies after the tomato crop, we grow some kind of leafy green vegetables like kale, and we could see that this was supplying nitrogen even beyond the tomato cover crop, tomato cash crop. So and that was reflected in the nitrogen mineralization that we measured. Soil phosphorus, we also saw an increased soil phosphorus levels with cover crops as well as organic amendments. So here you can see with crimson clover, normally you would expect the nitrogen to be higher, but we also saw a significant effect in phosphorus. So what we learned that during the cover crop, the crimson clover particularly can release an acidic root exudate, so that can really help in the mycorrhizal network that can promote soil phosphorus availability, and really the acidic exudates can break down the phosphorus that is associated with minerals such as iron or aluminum or calcium. So that is being put in the systems and made available to the cash crop. So we could really see that happening, and this was reflected when we looked at the leaf tissues and also tomato nutrient profile. We were seeing a higher phosphorus in the system. Likewise, with potassium, we could see higher potassium, especially with triticale and amendments. So this is really a summary of different time points. So, but when we really compared this against whether or not our crop or tomato crop was uptaking or accumulating potassium, we could see that, especially with our wheat middlings and cover crop treatments, there was an increase in potassium levels, especially in the above ground plant tissues. So here we are sort of looking at everything into a soil fertility score, looking at different soil fertility indicators. And we could see that really with the cover crop as well as with middlings, the nutrient supplying capacity of the soil overall was much higher. It was also comparable when we had only cover crop or when we had wheat middlings in the system. So we could see that throughout different sampling time where we did measure our soil fertility and different parameters, we could see that constantly uh, amendment as well as cover crop improved the overall nutrient holding and supplying capacity of our soil. When we compare different cover crops, again, kind of like following a similar pattern, we could see that with whenever we had cover crop in the system as well as wheat middling in the system, it was much higher than a fallow plot. And especially we could see that with crimson clover and organic amendment as well as triticale, even though there was some early nitrogen tie-up, but overall we could see that both triticale as well as crimson clover with middlings had a higher soil fertility score throughout the AST and post AST sampling times. Finally, here we are looking at tomato marketable yield, and we are seeing that compared to no wheat middlings, we could see uh, some increase, but not significant difference in the first year. But after the 2 cycles of AST, there was a significant increase. This was about a 40% increase compared to no wheat middlings in the second second year in 2024. So we could see that over time, really the organic amendments was building the soil fertility and soil health that was being reflected in our yields. In terms of cover crops, we didn't see, again, a significant effect, especially, but we did see that in the triticale cover crops, our yields were not as much as we expected. And this was probably because of the delayed start that they had, because of some of the nitrogen tie-up that we mentioned that happened uh, earlier in the season. And over throughout the first year and also the second year, we could see that the crimson clover treatment had the maximum yield compared to the other treatments. So it was showing up higher yields whenever we had crimson clover cover crop. So what are some of the things that we have learned and we continue to learn in our study? So we can, I think, safely say that based on our experience that whenever we can incorporate cover crops and organic amendment, this is really enhancing the anaerobic soil disinfestation in terms of a bunch of things, especially when we look at the soil organic matter, active carbon, nutrient dynamics, soil chemical buffering, and all those kind of things. We could see that compared to a control, we could see that whenever we had cover crop and when we supplement even that cover crop with organic amendments, it was helping to build overall soil health over time. In case of soil organic matter, we saw even increase compared to baseline. And in terms of soil fertility, whenever we looked at soil samples and also whenever we measure tissue nutrient assessment, we could see that cover crops as well as organic amendments really stood out when it comes to soil fertility indicators as well as soil health indicators. So by adding cover crops, we are adding living roots. We are providing soil cover. So this is reflected in our soil organic matter and fertility. But we are still using tillage to incorporate these cover crops, and this is why we are seeing the aggregate stability not as being built as much, even though compared to no amendment or no cover crop, we do see an increase in soil aggregation. But if we tease out the particular timing of sampling, we could see that as soon as we till the cover crop into the system and then we start to lose those soil aggregate stability that we gained with cover crops. So that is something we are trying to work with. So could we incorporate cover crops without tilling? Could we just roll and crimp the cover crops and supplement with other amendments? Could we apply biostimulants, biochar, all those other amendments that can help build aggregations with the cover crops? So that is something we are going to explore in the coming years. Overall, what we found is that high carbon to nitrogen inputs, especially the grasses such as triticale, help to build soil structure, but it delayed the nutrient supply, particularly in the early stages, that tied up some of the nutrients, which slowed the early growth. And that was sort of what we believe is reflected in a lower marketable yields of tomatoes over the years. Lower carbon to nitrogen inputs, this is crimson clover, for example, provided the nitrogen, increased the phosphorus also, as we saw, and it provided more in terms of soil fertility, but not as much in terms of building soil health over time. So what we really think is that when we combine the grasses with the legumes, I think that is when we can see the maximum impact, not only in terms of soil health and fertility, but also in terms of uh, managing those weeds that we saw. There were higher weeds in our crimson clover only or a legume treatment compared to a mixer where we had a good control of weeds, but also we had sort of more balance in terms of carbon that we are supplying, the soil fertility, and really the soil health that we are building. Could we grow other cover crops, or could we like add more types of cover crops in the mixes? For instance, as we didn't have any brassicas in the mix, so could that really impact the soil aggregation? That is something that we also plan to investigate in the future. So can we sort of have a more wider mix of cover crops that can be helpful compared to only grasses and legumes? So and that would also help us to maximize diversity in this system and feed into building a more resilient systems. So coming back to the overall soil health, just to summarize this, we do see that with cover crops, especially when combined with middlings, there was an increase in soil health over time, but our values are still sort of lower compared to around 30, which is considered more healthy soil in intensive vegetable production systems. So we'll continue to look into this, and we'll see in next year's samples. So are we, let's say, constantly improving the soil health, or we need to sort of supplement with other complementary inputs to really build, or we need to reduce tillage, or we need to find alternative ways to incorporate cover crops. So these are some of the different things that we are continuing to monitor and investigate in the future. So that will be an update that we'll provide in the coming year. Like I mentioned already, really wanted to emphasize this one more time because what we saw was that not only the cover crop choice, but also terminating the cover crop timely and supplementing with a labile organic amendment was really providing an optimum carbon to nitrogen ratio that was helping the soil health build up and also providing enough soil fertility for the tomato crop over the season. Like I mentioned already, so we are currently sort of looking at ways we can incorporate cover crops without having to till too much, or sort of bringing more type of cover crop in the mixes, or opportunities to combine cover crops with other amendments such as biochar. We are also looking at maybe biostimulants, bioinoculants, humic acids, different organic amendments. So we're kind of like really looking into different systems management so we can build overall soil health in the system. So that will be something we'll be continuing to do and report in the future. So with that, I would like to acknowledge especially the funding from the OREI project from USDA NIFA. So it was the ASD Easy Organic project, and it was a multi-institutional and multi-regional project. So we had collaborators from USDA areas, University of Florida, Penn State, and Oregon State University, and there was other collaborations as well. So, and a lot of this work was done by people who were at the Joya lab before I arrived. So I would like to acknowledge that a lot of the dirty work or the hard work was done by people, and I was mainly looking at the data and really looking at the soil health from this study. So with that, I think we have time for questions, I hope.
We do. And
Okay.
if anybody has a question, feel free to just type it into the Q&A box. I know there's a couple in there already, and we've got um, we've got at least another 10 to 15 minutes for questions. So
Uh,
thank you very much for that thorough presentation. And I just want to let anybody who didn't hear the very beginning that we recorded this presentation. You'll be able to find it on the eOrganic YouTube channel and also on the website for the project, which is eorganic.info/ASDEZ organic. So what was the rate of ASD application that you used?
uh, you mean the amendment? Yeah, I think it was around 15,000 kilogram per hectare was the rate that we use in this study.
Okay. And how long do you wait after mowing the cover crop to apply ASD, and how long do you wait to plant the major crop or the cash crop after ASD?
Yeah, I believe we incorporated the cover crop and applied the AST sort of immediately on the same day. So we didn't wait for after incorporating the cover crops, but after re-aerating the soil, we typically waited maybe a few days before applying the main crop.
Okay.
I'm not sure exactly how many days. Maybe Francisco can fill me in with that, but uh, yeah, typically we don't need to wait like too long before going into the main crop. So a few days is enough.
Okay. Let's see. Does anybody else have any more questions? We have a little bit more time. Oh, yeah, here's one from Deanna. Have you developed strategies for perennial crops like raspberries that would not require clearing the field?
Yeah, that is yeah, that is something we discussed and are discussing with the growers, like how can we use it for more perennial systems. I think what we did in this case was to make sure not just like go once, but we took the sample after we applied ASD and we waited at least 1 year to make sure that the nematodes were not coming back. So in the meantime, we encouraged the grower to grow a short-term cover crop. I think he grew some brassicas there, and then he grew some cucumbers before going into the perennial crops. So yeah, I think what I can say that if we sort of like proactively take samples and not wait until it gets like really tough, when we have to like when you cannot grow anything, we do you do need to clear everything, especially if it's like connected to the virus. Then there is no effective solution, I guess, other than like putting everything and dumping that and restarting again. Uh, Anything else that you wanted to add here, Francisco?
Yeah, I was gonna go back to the first question, the application rate for ASD. In this
Hmm.
case, the cover crop biomass is really depending on, the weather condition and
Yes.
like how much it rain, how long, when you uh, capitalize on that biomass. But for the wheat middlings, we use a standard rate, which is the equivalent of 6,000 um, kilograms per hectare of total carbon. But when it comes to pounds per acre, if that is a better number for you, the wheat middlings per se, like as it is, typically is relatively dry, is about 14,000 pounds per acre. Um, that's, based on the area that you are treating, if it is like just the raised bed or if you're applying to the entire surface, you can do the math there, considering that rate. Regarding the timing of application, like how long do we wait? Typically, we want to apply ASD in 3, 4 weeks, and we don't want to go longer than that just because growers want to establish their crop and, don't want to invest too much time in this treatment. You could do it for longer period, but, in that case, it really depends on the soil that you have. Like you don't want the soil to stay anaerobic for a long, long time. You don't need to. As long as you achieve enough anaerobicity to kill the pests and pathogens, you are good. So our experience is like 3, 4 weeks is good enough. And yeah, there's no reason to go over that. Regarding, strategies to apply ASD, while a perennial crop is there, I would say applying ASD while a crop is growing is not possible simply because the principle of this solution is the development of anaerobic conditions, and anaerobic conditions are not compatible with the growth of plants. So plants need oxygen to grow. So the timing, when you can apply ASD for those perennial crop is before you are establishing a new crop. And that's what we did with the grower that was affected by the virus. So the interesting part is that the virus was transmitted by dagger nematodes, which took the virus from weeds. As a mitigation strategy, like, if you have an issue, even an initial issue with dagger nematodes, the main thing you can do to protect your crop is to make sure that there's no weeds in your plot. So that's, like kind of a strategy there. And the grower, like even after applying ASD, we have recommended the grower to do that. And he's doing that, he's achieving that with using landscape fabric because it's an eye tunnel. In a different system, you will have to figure out something different. But yeah, I hope that addressed the question.
Great, thank you very much. Let's see, we have time for another couple questions if anybody has one. But if not, I just wanted to mention that I put the link to the ASD Easy Organic website in the chat box there for anyone who wants to learn more about the project or to see any previous webinars that we've done. We do plan to do another webinar or two on this project before it's over, addressing the microbiome issues perhaps, and then also definitely one on the economics of ASD. So looks like we don't have any more questions. So I just want to thank you, Jaya, for that presentation, and thank you, Francesco, as well for being online for the questions. And we'll look forward to additional presentations from this project as it progresses. So thank you very much.
Thank you,
Yeah,
everyone.
thanks, Elise. Yeah,
Okay.
thank you, everyone.